Assalamualaikum warahmatullahi wabarakatuh. I am RADZIAH and I am from class AJIS 24818. Today I want to talk to you about cloning process. So before we go to all about cloning, first we go to the what is DNA cloning. The cloning definition is making a genetically identical copy of an organism. Cloning can involve entire organism, single cell or even single genes. DNA cloning is making an identical copy of DNA. DNA cloning is commonly used in medical research, both to create treatment and drug, as well as to research disease states. Secondly, we go to the cloning process. The DNA cloning process involves insulating the genes of interest and cutting it with restriction enzyme. The same enzymes are used to cut a vector, such as a bacterial plasmid. A bacterial plasmid is a small circular piece of DNA that can be cut and pasted to include genes of interest and then inserted into bacteria to produce the protein product of the genes. After both the genes of interest and the plasmids are cut, are passed together, and then the plasmid can be inserted into the host bacteria. Sometimes different types host cells are needed to clone DNA. This cell can also be used with a vector called a AJIS, artificial chromosome. Even mammalian cells themselves can be used as vector depending on which genes need to be expressed. This is the process of gene cloning, but there are also other types of artificial cloning that include gene cloning, reproductive cloning, therapeutic cloning. Reproductive cloning use the same technique described as I said, except they are used to modify stem cells that can be used to create entire organism. This can be used to create organisms for human consumption, recreational use, or in medical research. Therapeutic cloning also introduces foreign genes into stem cells, but instead of creating an entire organism, the cells themselves are used for research or to replace damaged or diseased tissue. So next, we go to step in gene cloning process. How to clone a gene? The gene cloning there are as 3 steps. The number 1 is isolate the genes of interest. The first step is to isolate the genes of interest for cloning. This can be done by isolating the DNA from the cell that contain it and cutting it out with specific restriction enzymes. Restriction enzymes are bacterial enzymes that cut DNA at specific sequence. Number 2 is plasmid insertion. The plasmid of choice is then also cut with the same restriction enzyme. This creates sticky ends that overlap and can be stuck together, allowing for insertion of the genes of interest into the plasmid. The genes of interest and plasmid are then stuck together using the enzyme DNA ligase. So the last step is plasmid transfection. The plasmid is then inserted into bacterial cells throughout the process of transfection. Generally, the plasmid will contain a selection marker, such as something for antibiotic resistance or the ability to metabolize a certain sugar to produce a color. This allows scientists to select the bacteria that were successfully transfected. Then we go to how the cloning process happens. Cloning is the process of making a genetically identical copy of a gene of all organism. Cloning is done using molecular biology technique. First, the genes of interest is cut and pasted into a vector, such as a plasmid, to form recombinant DNA. The recombinant DNA is then inserted into her host, which make copies of the genes. Next, we go to benefit of cloning process. Benefits of cloning include being able to create tissue and organ that doctor can use when needed for surgery on the original. If lab can clone and grow only the part needed, they would eliminate the moral and ethical issue associated with cloning an entire person. Other benefits include growing stem cell, cloning lab, might genetically engineer for the specific study, bringing back extinct species, reproducing a pet that died, and cloning livestock for food. Besides, we have the advantage of cloning process. Number one is cloning does not need to involve making a whole new person. Imagine if a person has a failing liver, what if the cell of the liver could be cloned so that a new liver could be created and then transplanted? It would be an easy way to solve the organ scarcity issue that currently exists. The process of cloning could also be used to repair or grow new cells to replace damaged or missing ones, which could treat illnesses and genetic disorders. The number two advantage of cloning is it removes the barrier of infertility. Because cloning uses adult somatic cells, it is a process that allows anyone to have a child that is biologically their own. Even if that person has a reproductive system which does not support fertility, doctor could take the somatic cell and implant them into an embryo, creating new life. This technology will give everyone the chance to become a parent, even if they were not sexually active. The last one or advantage of cloning is it could extend human life capabilities. In the developed world, the average lifespan is approaching 85 years for top nation. Even in the United States, the average lifespan is upward of 70 years for men and women. Not only could cloning help to extend life to ever longer lengths, it could be a way to bring the rest of the world up to the current standard as well. In Sierra Leone, for example, the average lifespan for an adult male is just 49.3 years of age. Furthermore, we also have the advantage of cloning. First is the result on society would be unpredictable. The most common arguments against cloning involve the unknown that would happen to society. If parents would be able to manufacture children to specific genetic profile, then there is the possibility that genetic variation could decrease. This would result in humanity becoming more susceptible to disease and deformity, requiring more genetic selection because we would eventually be inbreeding with ourselves. Number two is the rich will get richer and the poor will disappear. A society where genetic selection is possible will place a higher emphasis on socio-economic means of each person or household. Those who could afford cloning would essentially create their own class, while those who could not afford the process would likely be shunned or disregarded by the rest of society. The last one is it is an unpredictable and certain process. Cloning is far from a perfect science. Many of the disadvantages involve the what if of this science, but there are some facts to think about too. When Dolly was successful clone, only 98 out of 300 were successfully implanted with adult somatic cells to create a pregnancy. Out of those 98, only one was successful delivery to term. Although advances have been made since Dolly in the field of cloning, the science still has a long way to go. last but not least, I would like to conclude that cloning can help in numerous situations such as medical emergency, reversing heart attack, treating genetic defect, leukemia, infertility, lowering transplant rejection, and helping endangered species. Although cloning can save many lives, it is deemed unethical and should only be used in this situation. It should not be used for fun or experiment that will not affect the world positively. In the future, cloning could be used to find a cure for multiple disease and helping endangered species. So that's all about cloning process from me, and I hope that you all can understand about cloning process that I taught just now. Happy watching everyone, and before I forget, at the end of the video I will attach the DNA cloning process and genes cloning process. So have a nice day and have a good day everyone. Thank you.